Thank you, and good afternoon. Thank you for the opportunity for letting me present. I have no relevant conflicts of interest to disclose. The purpose of the study was to characterize the aqueous humor in primary open-angle glaucoma patients and correlate the findings with disease severity. So overall, we had three groups: controls without glaucoma, mild glaucoma, and advanced glaucoma. The idea was to sample aqueous humor from all these three groups with an N of 10 in each group. After getting ethics board approval from the two participating centers, informed consents were obtained from all participating patients. And the staging of the glaucoma was based on the mean deviation values. The aqueous humor collection was done with a 30 gauge needle mounted on a 1 ml syringe, and 100 microliters of aqueous was withdrawn from the anterior chamber. The glaucoma samples consisted of primary open-angle glaucoma eyes, mild and advanced disease. Control samples eyes did not have glaucoma. We excluded patients who had prior ocular surgery and those with ocular inflammation. After collection of the samples, these were frozen on dry ice and then stored at -80 till the final analysis. The final analysis panel had about 8,000 proteins, including cytokines and other mediators. The test that was used to analyze these samples was a glass slide-based antibody kit, and the aim was to analyze 8,000 target proteins, including mostly cytokines. These are the demographics. We had 10 eyes in each group, and the age and the sex were equally matched between the groups. And the results showed that there were a significant number of differentially expressed biomarkers with a false discovery rate of less than 0.1, the most being in the advanced versus control group. And it's very important in such large data sets not to take cytokines or individual mediators in isolation, but to consider them as a whole to look at any possible pathways that are standing out. So with an advanced to mild comparison, we found that multiple pathways related to immune activation were upregulated. In a mild versus control comparison, pathways related to vascular development, cell migration, and decreased proteolysis was upregulated. Now, this is a cross-sectional study, but we can, and ideally a longitudinal study is required to really say that this is linked to glaucoma pathogenesis, but we can extend and say that these may be involved in glaucoma pathogenesis. In an advanced to control comparison, there were several factors that stood out, including angiogenic factors such as angiopoietin, pro-inflammatory cytokines, immune modulators, and structural proteins such as matrix metalloproteinases. Now, just a brief discussion. We found that IGF-1R, the IGF-1 receptor, was significantly increased in advanced glaucoma, and this has been implicated previously in neovascularization and in thyroid eye disease, and this may be a new target that can be studied in glaucoma pathogenesis. Similarly, angiopoietin-1 recently was shown to be really involved in the formation of Schlemm's canal and its integrity, and knockout models had significant glaucomatous changes. That's another gene that can be studied in glaucoma pathogenesis. In conclusion, this study has identified clearly many pathways that are implicated in POAG pathogenesis as well as progression. Now the next steps would be to validate these findings. So this can be seen as a large study that is a starting point to further targeted approaches. To validate these studies in animal models will help us identify further mechanisms, especially with relation to vascular markers and angiopoietin. To rule out the effect of glaucoma medication, because many of these patients were on glaucoma drops, this can be done using in vitro techniques using TM cells and using qPCR to see if these same genes are upregulated, and to correlate the findings with clinical characteristics as well as OCT and visual field parameters. Thank you, and I'd like to acknowledge the funding sources and my team. Thank you.
Thank you, Dr. David. How do you plan to take this study forward? Like, what would be your next step of research in this aspect?
Yeah, one is to, so this has given us a lot of pathways to work on, especially vascular markers such as angiopoietin and other, especially there are other parameters related to lymphatic formation. So the next step will be to isolate these findings in animal models so we can create knockout models of these genes and study that specifically. And the second, I would say, is to really study the effect of glaucoma medication, because that can affect the cytokine profile in that aqueous humor. So to really make sure that this is not an effect of the treatment that we are seeing, that's another step.
Thank you.